Hello, this is video 9 out of 10, and at the end, if you have time, I would like you to watch the C'est pas sorcier maladie génétique. And today's lesson's about genes, heredity, and genetic variation. Let's consider the genetic variation of the students in this class. So take a look around and name 2 traits that show non-genetic variation in the students. These would be features that are not inherited. I want you to also name 2 traits that show genetic variation in the students in this class. So pause the video now and discuss. And now, did anyone say hair length or style of hair for non-genetic variation? Hair length is something that we control by cutting it. Our genes do not control it. Did anyone say height for genetic variation? That is true, but it can also be influenced by nutrition, which would not be genetic. Remember the macromolecule nucleic acid that forms DNA? We will examine it in more detail. Here is a chromosome, which is made from a strand of DNA, and thus contains the codes or the genes that determine skin and eye color and other traits that we inherit from our parents. Our chromosomes come in pairs, one from our father, one from our mother. On each chromosome, we find genes for hair color and eye color, and these genes could code for different things, different colors, depending on the mother or the father. We have not been able to see the structure of DNA until the mid-1900s, maybe around 1950. But in 1822, a monk was born. His name is Gregor Mendel, and he studied how traits are inherited. He was an Austrian monk who studied plant breeding and mathematics. His experiments with pea plants became the foundation of modern genetics. Mendel created pure lines of peas and hand-pollinated them to control the lines of the mother and the father plants. How did he do this? Well, on a flower, we find stamens that release pollen. Here are the stamens. And there's also a carpel that contains the eggs. Remember, we learned about that when we learned about angiosperms. Plants that are self-pollinated have pollen that fertilizes the eggs on that same plant. And plants that are cross-pollinated, pollen fertilizes eggs on a different plant. Mendel controlled this by removing the stamens so that the flower could not pollinate itself. He then used a brush to transfer pollen from a white flower, which was the father, to the carpel of a purple flower on the mother plant. Thus, he controlled the genetic lineages of these plants to get true breeding varieties that always produce offspring identical to the parent. For example, purple flowered plants produce offspring with only purple flowers. White flowered plants produce only white flowered offspring. he crossed these true breeding flowers and grew the resulting peas. The resulting peas are the hybrid plants. And these hybrids had only purple flowers. A hybrid is the offspring of the true breeding parents. Mendel went a step farther and crossed these hybrids with each other. And he got the strange result of 75% purple plants, 25% white. Mendel used the scientific method to investigate this phenomenon of white flowers coming from purple parents. He formed several hypotheses that he could test. We will examine each in the following slides that I would like volunteers to read. Then join me at slide 11. We learn about dominant and recessive alleles that are inherited from our parents. Remember that a character is an inheritable feature like flower color. A trait is the version of that feature, like a white flower or purple flower. Another character is position of the flowers on the plant. Dominant is axial, meaning the flowers come from the axis of a leaf. Recessive is terminal, meaning the flowers are on the tip of the plant. The character of the color of the seeds, we can have yellow or green, and the shape of the seeds, round or wrinkled. Mendel's fourth hypothesis, which we will call the law of Mendelian segregation, states that a pair of alleles separates from each other when gametes are made so that an egg or sperm has only one allele. Thus, an egg or a sperm gets only one of the two alleles that are present in the somatic cells of the organism making the gamete. And someone define what somatic cells are and what are gametes. Here is that hypothesis illustrated. Remember that Mendel didn't know about chromosomes, so this wasn't even proven until 100 years later. Somatic cells are all the cells in our body, except for the sperm and the egg. We will learn this in more detail in the next unit, but here is a summary of how gametes form. It is called meiosis, and it occurs when one somatic cell, which is a body cell, divides and forms 4 gametes. 1, 2, 3, 4. Gametes are either eggs or sperm. The pair of chromosomes in each somatic cell also separates. So these 2 chromosomes separate, and then each chromosome divides so that each resulting gamete has only 1/2 of a chromosome. Remember that the alleles, or each version of a gene, are found on the chromosomes. Thus, the pairs of alleles are separated from each other when gametes are made, so that an egg or sperm has only one of these alleles. And here's allele for white flower, here's the alleles for purple. We can say the alleles are segregated. Remember that the somatic cells of an organism has two alleles, 1 and 2. So in this case, this plant cell has an allele for white flowers and an allele for purple flowers. Here are the various ways to note the results of inheritance. Remember that the character is the color of the flower. The trait is a violet flower or white flower. So here it is illustrated, but we can show the genotype, which refers to the trait determined by the genes. So in this case, we use V for violet. Big V is dominant. Small v is recessive. That's genotype. However, phenotype is what we can see. It's the appearance. So we see the purple color, it's present, or we see the white, meaning purple is absent. Remember that genotype refers to the genes. A purple flower can have two purple genes or alleles. Or it could have one purple allele and one white allele, and it would still look purple. The phenotype is what we see. It is the color. We know a white flower has only the recessive alleles for white. If it had a purple allele, we would instead see purple. But here we have two small v's, which show recessive genes, and the phenotype is what? White. We've got specific terms for the big V and the little v. Homozygote. Homo means the same. So two of the same alleles is homozygote. The white is a homozygote. Dominant purple is homozygote. Or we could have a heterozygote, in which we have one of each. We have the same, so it's homozygote. These are different, it's heterozygote. So I would like a volunteer to come up and explain P, color, and shape. Another notation you need to know is generation F. It's the first generation from the parents. Generation P is parent. Generation F1 is the first generation. In this case, parents that were true breeding to either purple or white produced purple flowers. What percentage of the offspring are purple? 100% are purple. They have a big V and little v for their alleles. This is a Punnet's table that allows us to predict percentages of offspring of each trait. In this example, we look at true breeding seeds of yellow or green. Which color is dominant, and how do you know this? We know it is yellow because it has a big J. So we take the table, we put the dominant across here, put the other type across here, and we can fill out each square by following this across. So this first one, large J, little j. We follow this across, large J, little j. Do that for each one. What's the result? 100% have yellow seeds. And Elio might want to stop and explain that again. Let's go back to flower color from crossing the purple flowers of the F1 generation. Remember, F1 had large V, little v, which means that we see the color purple. Note these questions along the side. We'll be able to answer this once we fill out the table. I want Elio to pause this and for someone to come up front and explain each part of this table and fill it out. Is this what you got? Please pause the video again and discuss how you calculate these numbers. We can also fill out a Punnet's table for more than one trait. Here we see both color, yellow or green, and form, smooth or wrinkled. Pause the video and discuss this. Then the following slides I would like volunteers to read and discuss. So pause this and discuss, and then go to each slide and discuss. Now let's discuss genetics on a population level, rather than just parents and offspring. What is a population? It is a group of individuals of the same species that live in the same area and produce fertile offspring. Fertile is very important, or else the species would not continue. Here we have an example with cattle egrets, or Crabier Gade-Beef. We see them all over Haiti. Here's one in central Haiti, a population in central Haiti, population in southern Haiti. They are the same species. A egret from central Haiti can breed with an egret from the south and have fertile offspring. However, genetics of populations change. It is very far to fly from central Haiti to the south, so they don't often breed. Over time, their genetics can change so that the birds begin to look different, and we can even have infertile offspring, in which case these are two different species. We have an example of this in the Catje tête noire. What color are the heads of the Catje tête noire in the north? They have a black crown. The ones in the south have a gray crown, and they are different species. How do populations change? Here's an example with beetles. In the beginning population, we see that some beetles have the allele for brown color. Pause this video and discuss why there are no green beetles in this final population. Then read and work through the final slides as a class or on your own as time allows. Finish with the C'est pas associé genetic maladies video if you have time, and I will see you at video 10. Thank you.